Hi everyone. Um, in this video I'd like to start our discussion of sex determination. And I'd like to kick off our discussion by talking about switches. And so the main points of this video will be that switches that give two very different regulatory states are common in gene expression and that they commonly make use of positive feedback. Okay, so let's jump in. So we've talked about a variety of different gene regulatory problems or functions or challenges that are solved. So what I mean by this is we've talked about a case where lactase is regulated in a stage-specific manner in mammals, on in adulthood, and then on in childhood, and then off in adulthood. We've talked about environmental condition-specific regulation, in which E. coli senses lactase and glucose in the environment and responds with a regulatory program, either expressing or not expressing lactase. We've also talked about tissue-specific cases. So actually the clearest one here is NOVA, and we noted that NOVA is expressed or is on in the brain but is off in other tissues. But we also talked about genes involved in formation of pigmentation, which it turns out are also expressed only in certain tissues. And finally, we talked about the challenge of moderating the level of product. We talked about how ribosomal protein coding genes negatively regulate themselves in order to avoid either too low or too high of an expression level. And so our question then is, what is the main challenge in sex determination? So this is slightly different, right? In this case, we're not talking about being able to, an organism being able to switch regulatory program in the middle of a life stage or in response to environmental conditions or across different tissues. In this case, sex determination is largely intending to or ideally producing two different states, fertile males and fertile females from two permanent states, right? So that this is a differentiation across individuals where individuals are to have either a regulatory program A or regulatory program B. So what we're talking about here is kind of a switch, right? We want to switch some way for the organism's genes to make decisions between two different possibilities and to keep those possibilities stable. And so this is a paper from my friend Josh Cherry, and this is his little joke from The Wizard of Oz, are you a good switch or a bad switch? About how exactly to make a biological switch. So it turns out that if we look at such cases where we're talking about regulatory switches between two different states, permanent stable switches, that we see that a common mode is positive autoregulation. So let's remind ourselves of what autoregulation is. So we've studied negative autoregulation. We talked about a case where a gene makes a product A that binds to negatively regulate itself. And we said that if we think this through, under these circumstances, when there's no product A, then the gene will not be silenced, and so it will produce gene product A. As gene product A accumulates in the cell, some of it will come and negatively regulate gene A, therefore shutting off further production of protein A. So what this means is that we expect, if we start at a low level, that the gene product will come to a moderate level. It also actually means if we start with a very high level, that the gene will be silenced, and over time, protein product or gene product will be degraded, again getting us back to a moderate level. And we talked about the case of ribosomal protein coding genes, which actually regulate themselves at the level of splicing. So if there's not much ribosomal protein coding gene around, transcripts get spliced, and those spliced transcripts are translated, giving rise to ribosomal protein coding, ribosomal proteins. As these accumulate in the cell, they will bind to transcripts and cause preferential non-splicing of the transcripts, leaving the transcripts unspliced. And these transcripts, it turns out, are degraded, thus giving rise to no more protein. This is another case of negative autoregulation. So what about positive autoregulation? So we have here our gene B, and we note that gene B has a binding site for gene product B, or protein B. So in this case, if we have no protein, well, there's no protein to come over and bind and stimulate expression of gene B. So in fact, if we start with no protein, we'd expect that we'll just stay with no protein. However, if we start with a moderate level of protein, then some of this protein can come and bind to the binding site and stimulate expression of gene B, giving rise to more protein. And the more protein we have, the more protein we have to bind and stimulate transcription and expression of gene B all the more. So what we've said here is that if we have no protein, then we're going to stay stable with no or very little protein. Whereas if we start with a moderate level of protein, that's going to accelerate and give us a very high level. That we have two states of low and high. Exactly sort of what we're thinking about in terms of making a switch, that it's either on or off. So let's have a look at the genes involved in sex determination in vertebrates and have a look at their relationships. So it's a little bit more complicated here. It's not just a single gene regulating itself. So it's in fact in large part a two-part switch. So one gene called SOX9 produces protein, and that protein binds to the promoter of FGF9, stimulating production of FGF9 protein, which then binds to SOX, stimulating more binding, stimulating more production, stimulating more binding, and so forth. So we can see that just like a simple positive autoregulatory circuit, in this case, this is going to have what we call positive feedback. Right? So this is not autoregulation because auto means self, and the genes are regulating each other, not themselves. But we can more generally say this is positive feedback. And what we mean by that is if there's less of a gene product, then we expect that to lead to even less of that gene product. If we start with more of a gene product, we're going to get more. This is the poor get poorer and the richer get richer. Okay, so we can write that in our regulatory language this way, right? SOX9 positively regulates FGF9, FGF9 positively regulates SOX9. Okay, so we sort of argued that once we get things rolling, we expect both, once we have a little bit of protein expression, we expect both of them to go to high levels. But we might be concerned that things could be a little bit noisy, right? So what happens if SOC9, just by imperfection, starts expressing a little bit of extra protein, or a little bit of protein? Well, that could have a positive feedback, and we get more and more and more. So to make this switch stable, there's actually a third member of the switch, which is called Wnt4, and Wnt4 negatively regulates SOX9. And in addition, FGF9 negatively regulates Wnt4. Okay, so let's think about how this is going to work. If we start with a moderate level of Wnt4 protein, that's going to bind and repress SOX9. It's going to turn SOX9 off. Therefore, SOX9 will not stimulate production of FGF9, and FGF9 will not suppress Wnt. So we expect to get more Wnt and stably high levels of Wnt and stably low levels of our other two genes. Okay, what about the other way around? What if we start with moderate levels of SOX9 and FGF9? Well, we expect FGF9 will come and shut off Wnt. Consequently, Wnt will not bind and suppress SOX. However, FGF9 and SOX will bind to their corresponding binding sites and stimulate production of SOX9 and FGF9. So we expect to get stably high levels of SOX9 and FGF9. Okay, so we can add this to our regulatory diagram. So this note that we showed that FGF9 regulates Wnt4 and Wnt4 regulates SOX9. And so here is our core relationships in this switch. And we worked out that we expect under these circumstances to go to either one state where we have a lot of SOX9 and a lot of FGF9 and little Wnt4, or alternatively to go to a state where we have a lot of Wnt4 and repression of the other two genes, leading to a lot of Wnt4 and little SOX9 and little FGF9. Okay, but I haven't told you which is which, right? I've argued that we have these two stable different states, but in the instance we're talking about sex determination, so which is associated with which? So across a wide variety of vertebrates, what we see is that the female state, the regulatory state common in females, is to have Wnt4 at high levels and the other two at low levels, and the male is the other state. The male state is the other state with high levels of SOX9 and FGF9 and low levels of Wnt4. So our next question that we'll cover in the next video is, so what kicks it all off, right? We basically convinced ourselves that this can go one of two ways, but there needs to be some kind of control to determine which of those two stable states we will arrive at. So that will be the topic for our next video, but let's just remind ourselves we talked about switches with two very different states are common in gene expression, and we noted that these switches commonly make use of positive feedback, and we went through the core switch for sex determination. Thanks very much.